Hi everyone and welcome. My name is Eric Azúcar Lopez and I will be doing a brief overview of glycolysis. This instructional video is for my biochemistry class Chem 40420 at Notre Dame University. Today we're going to walk through one of the most essential processes in cellular biology, glycolysis. This is the very first step in cellular respiration, the process by which cells break down glucose to make energy. We'll cover what glycolysis is, why it's important, and go step by step through the pathway in a way that's easy to understand. So, what exactly is glycolysis? Glycolysis is the process of breaking one molecule of glucose, as seen over here, a 6-carbon sugar, to two molecules of pyruvate, as seen over here. This process happens in the cytoplasm of the cell, and it does not require oxygen, so it can happen in both aerobic and anaerobic conditions. Along the way, the cell gains a small but crucial amount of energy in the form of ATP, which is the cell's energy currency, and NADH, which will be used later to make even more ATP. Now, let's take a high-level look of what goes in and what comes out of glycolysis. Here are the inputs, and we can see that glycolysis uses one molecule of glucose, two NAD plus molecules, two ATP molecules, and 4 ADP molecules. And for the outputs, we can see that glycolysis generates two pyruvate molecules, two NADH molecules, and a total of 4 ATP molecules. However, it is important to know that glycolysis only makes two net gain ATP molecules, as two of the two molecules are used initially in the entire process. Now, let's go deeper and break glycolysis into its 10 different steps. I will be grouping these different steps into two phases, starting with phase 1, which will be the investment phase. In this phase, the cell spends 2 ATP to make the glucose more reactive. In step 1, which can be seen right here, glucose is phosphorylated by hexokinase using 1 ATP. This forms glucose 6-phosphate. In step 2, glucose 6-phosphate is rearranged into fructose 6-phosphate by an isomerase enzyme called phosphoglucose isomerase. In step 3, a second ATP is used to add another phosphate, forming fructose 1,6-bisphosphate. This step is catalyzed by phosphofructose kinase 1, which is the main regulatory step of glycolysis and can be found in step 3. After this, in step 4, the 6-carbon molecule is split into two 3-carbon molecules, one of them being glyceraldehyde 3-phosphate and the other one being dihydroxyacetone phosphate. These two molecules are made by the enzyme aldolase. In step 5, dihydroxyacetone phosphate is converted into another G3P molecule. So now we have two identical G3P 3-carbon sugars, and this is through the enzyme called triose phosphate isomerase. It is important to understand that step 1 and step 3 of the investment phase in glycolysis are both irreversible steps. Now let's take a look at the next 5 steps of glycolysis, which are part of the energy payoff phase. In step number 6, each G3P molecule which was made previously is oxidized and phosphorylated, producing 1,3-bisphosphoglycerate and reducing NAD plus into NADH with the use of the enzyme G3P dehydrogenase. In step number 7, each molecule donates a phosphate to ADP, making ATP and 3-phosphoglycerate. This is called substrate level phosphorylation, and it is done through the enzyme phosphoglycerate kinase. In step number 8, the molecule 3-phosphoglycerate is rearranged with the use of phosphoglycerate mutase, and it turns it into 2-phosphoglycerate. In step number 9, enzyme enolase turns 2-phosphoglycerate into phosphoenolpyruvate, a high-energy molecule, releasing water as a byproduct. Step number 10, pyruvate kinase transfers a phosphate from phosphoenolpyruvate to ADP, making another ATP and forming pyruvate. Since we started with 2 G3P molecules, everything in this phase happens twice, which is how we end up with 4 ATP and 2 NADH total, as explained previously. So, why is glycolysis so important? Glycolysis is the main way cells generate ATP under aerobic and anaerobic conditions. Glycolysis products feed other pathways. For example, if oxygen is available, pyruvate goes into the mitochondria for the Krebs cycle and oxidative phosphorylation, which produces more ATP. Furthermore, pyruvate can also be used for fermentation. In the case of mammals, this produces lactate. In the case of bacteria, it produces ethanol. Both of these processes produce NAD+, which can be used for step 6. Lastly, as a quick summary, I would like to wrap things up by summarizing what we've learned so far. Glycolysis is made up of 10 different steps, and it is important to remember that steps 1, 3, and 10 are all irreversible. Glycolysis makes 2-pyruvate, 2 ATP, but 4 ATP in total. It's a very important fact to remember, and 2 NADHs. Lastly, glycolysis is the gateway to more energy production. Okay, and right here, right under, I have some memes that I hope you guys can understand after this brief introduction or overview over glycolysis. Thank you.